is TSI slants. Now, TSI slants are very, very, very strange, but also very cool. They are differential, but not selective. So TSI slants are essentially tubes. In this slant, there's two pieces of it. You can see here we have this is a perfect example. This one's broken into the two pieces. You draw a line, you see the slant part. I'm going to outline that. Right here is called the slant. And then down here where it's broken off at, you see the butt. So the different color combinations between the slant and the butt is what you're going to use to diagnose the bacteria. So how does this work and what's going on here? TSI, triple sugar iron. Triple sugar, 3 sugars. There are 3 sugars that are found inside of this media. So this is a triple sugar iron slant with nothing growing inside of it. It's just a brand new slant from the manufacturer or out of the refrigerator after you made it yourself. You can see it's kind of an orangey color. So you're going to introduce some bacteria. You're going to stab them down inside of here all the way to the bottom of the little glass tube. And you're going to pull it up and you're going to streak the top. So you're going to put it in the incubator and you're going to come back 24 hours later, 18 to 24 hours, and see what happens. Now there are 3 sugars inside of here. However, there's the 3 sugars in this are glucose, lactose, and sucrose. Pretty much every bacteria on the planet can ferment glucose. Not all of them. Pretty much everyone can. What do bacteria produce if they ferment sugars? Acids. Not every bacteria can ferment lactose. We've talked about some that can. Not every bacteria can ferment sucrose. Some of them can, some of them can't. Those 3 sugars allow you to tell apart what's going on inside of here. There's also 3 different concentrations of sugar inside of it. There's a little bit of glucose, a lot of lactose, and a lot of sucrose. So what's going to happen is you introduce some bacteria to this tube. If they can eat glucose, they're going to eat glucose very, very, very fast, very quickly. And what's going to happen is when you eat that glucose, the glucose is going to be found all the way throughout the tube, very simple sugar to break down. You're going to produce an acidic waste byproduct. You break down sugars, that's just what happens. Fermentation of sugar produces acidic waste. You break down that waste, that sugar, and the entire tube, Phenol Reds, the indicator in here works like our mannitol salt agar. Phenol Red is going to turn bright yellow in the presence of our acid. The entire tube will turn yellow. Yellow slant, yellow butt. And that's what's going on here. Very quickly turn yellow. This is red as the slant over the butt. Yellow slant over yellow butt. The combinations, colors of the slant and the butt is what you're going to look at to diagnose your species of bacteria. Yellow slant over yellow butt. Able to ferment glucose as well as, so this is what it would look like. Now, so if you let this guy sit for about 5 or 6 hours, the glucose is going to run out. And that's the key point here. That's what we're looking for. Can they eat another sugar? If you can eat glucose, that's fine, cool. Pretty much everybody can. What we're looking for is what else can you eat? So what's going to happen here? This guy right here, poor little Pseudomonas, could not break down glucose. He could not eat anything but proteins, so he didn't change at all. He produced a red color. He broke down the proteins. Hydrolyzed proteins produced a basic waste byproduct. That basic waste byproduct came in contact with the phenol red indicator and produced a red color. Red on top, red on bottom. Red over red. Red is referred to as the letter K. K stands for alkaline. K over K. Red slant, red butt. K over K is how this is reported. That's how you would read this. Alkaline butt, alkaline slant over alkaline butt. This guy was not able to ferment any of the sugars found inside of this media. None at all. Now let's move over here. This guy has a red slant over a yellow butt. Let's start with what would happen with with incubating this guy from the get-go. You slant him, you streak him, he looked like this from the get-go. You put it in the incubator. 5 hours later, it was all yellow. The glucose ran out, and that's the point. We're looking to see if these guys, this particular species of organism right here, can eat anything other than glucose. Is it capable of eating lactose or sucrose? Well, this guy could not do that. He was only able to eat the glucose. It's the only thing he could eat. So you know he was able to eat the glucose because the entire tube turned yellow at one point. It's still yellow on the bottom. Protein fermentation, or hydrolysis, excuse me, takes best in the presence, or does best in the presence of oxygen. It's an aerobic process. So it doesn't do very well anaerobically. So this guy, the species here, 5 or 6 hours into his life, ran out of glucose. Instead of starving to death, he just started hydrolyzing proteins. He couldn't eat the lactose, he couldn't eat the sucrose, so he started hydrolyzing proteins. And that's pretty neat here. So you look at this guy and you go, oh, this bacteria is able to eat glucose, but he cannot eat sucrose or lactose. He started eating the proteins. So that's a good informational step to tell you what you're looking at to diagnose this particular species of bacteria, to figure out what you're looking at. It can eat glucose, but not lactose or sucrose. So how this particular tube would be reported here, the slant is basic, K. The butt is acidic, which is given the letter A. So K alkaline, K slant, over A butt, K over A. And that's how you would report the results of this particular TSI tube. Now over here, our yellow one on top, yellow over yellow, or A over A, acid over acid. This guy was able to ferment the glucose. We know that his entire tube turned yellow. 5 or 6 hours into his life, he ran out of glucose. This particular species of bacteria, whatever he is, was able to eat either lactose or sucrose or maybe both. And we know that because the slant remained yellow. Protein hydrolysis takes place in the presence of oxygen. He didn't need to do that. He was able to eat lactose. He was able to eat sucrose. He didn't need to break down the proteins. Given a couple of days or so in here, he might start doing that, but he didn't yet. So he was able to eat the protein, or eat the sugars. So we know this particular species of bacteria is able to eat glucose as well as lactose or sucrose, which is a very interesting bit of information that then you can use to diagnose what you're looking at. So this would be reported A over A, acid over acid. Another little caveat here, you can see this particular tube here is broken in half, and you can see here the presence of gas, plus gas. Some species of bacteria produce large amounts of carbon dioxide, methane, oxygen, lots of different types of gases through their products of metabolism, that breaking down those proteins and things, breaking down those sugars. And sometimes they produce so much gas that it gets trapped inside of the agar and it has to go somewhere. It can blow the agar apart. It can sometimes blow it out of the tube. There's so much gas that they produce. This one just broke apart the agar a little bit. Some bacteria species can produce gas. Some of them cannot. That's also a key characteristic of diagnostics. If you have A over A, or A over A plus gas. So if the bacteria is A over A, it's one species. If it's A over A plus gas, that's something else. That particular species of organism was not able to produce gas, but this one is. So that's something different. So it's another little bit of information that you can get. And gas production can occur in any of these, by the way. The next one over here, this is considered A over H2S, acid over hydrogen sulfide. Now, technically what occurred here is this is A over A, yellow over yellow. This particular organism was able to eat the sucrose, lactose, and glucose. The entire slant is nice and yellow. The butt is nice and yellow. But this particular organism is also able to produce a sulfide compound called hydrogen sulfide. There's this little bit of sulfur inside of this media, a little small sulfur source. Some organisms are capable of eating it. Some of them are not. And that's also a little bit of information that you can get. If you're a bacteria species that's able to eat it, that's something that's useful to know if you're trying to diagnose the bacteria found inside of your patient. Can they eat this? Does this species that they're infected with, is it able to eat sulfur or not? And then you can use that information to diagnose your patient and figure out what they've got. So this guy, this particular species was able to eat glucose as well as lactose and or sucrose, but it also is able to produce hydrogen sulfide. That hydrogen sulfide production occurs anaerobically in the lack of oxygen in the butt portion of the tube. you can see the dark black hydrogen sulfide is going to obscure the bright yellow of the acid butt. So you can't see it. It's there, you just can't see it because of the hydrogen sulfide. The black hydrogen sulfide covered it up. So A over A plus hydrogen sulfide is how you would report that one. A over A plus black hydrogen sulfide. This one over here, this guy was able to produce glucose. The bottom's yellow. Now you can't see it. Everybody's able to eat glucose for the most part. You can't see it. He ran out of glucose and had to switch to eating proteins because he couldn't eat our hydrolyze our proteins. So this tube would look exactly like this tube if you could see the bottom of it. So this is the exact same thing. Red slant, yellow butt. He was able to eat the glucose, ran out of glucose, could not eat sucrose or lactose, had to switch over to hydrolyzing proteins. That does best in an anaerobic or aerobic condition, which is why the slant is red. Once that happened, it turned the slant red at the same time that organism was producing hydrogen sulfide, which was going to cloud out the yellow color on the butt. So this would be reported K over H2S or K over A plus H2S. Depends on the book, and it depends on the professor in question. I don't particularly have a preference for these either way. So this is control with nothing on it. K over K, no change, NC over NC. Depends on how you want to do that. No change over no change, alkaline over alkaline. K over A, A over A, A over A plus H2S or A over H2S, and then K over A or K over H2S. It just depends how you want to report that, or K over A plus H2S, So what you guys would be expected to report for this one is if I gave you one of these, you could tell me if it's K over A or whatever it is. Tell me that these guys were able to eat the glucose but not the sucrose or the lactose and things like that. So once again, here's a YouTube video of someone going over this auger. Again, if you'd like a refresher on that or someone else other than me explaining it. So that's all I got for you guys on this PowerPoint, guys, for our media, very commonly used medias in microbiology labs and how they work. So if you have any questions on that, feel free to ask. Other than that, have a good rest of the day.